Unit 4, Part 1, Cell Communication and Homeostasis. Shout out to Mr. Austin, who has excellent notes, and we're going to go through this. So Unit 4 is cell communication and cell cycle, but Part 1 is about cell communication as well as homeostasis. It comes with this great note packet. You should be writing down your own information here while this is gone through. It's going to help you the most. And so here you can see it continues. You'll see diagrams that will help you. And continuing through the unit, here's where topic 3 starts. And then we get to our final topic of topic 4, okay? So we're going to go one by one here, and this is signal reception. So an overview of cell communication. Cell signaling is essential for both unicellular and multicellular organisms, and it allows cells to communicate with one another and respond to their environment. In single-celled organisms, cell signaling influences how the cell responds to its environment. In multicellular organisms, cell signaling coordinates the activities within individual cells that support the function of the whole organism. Similar pathways and molecules used for cell communication are shared amongst all organisms on Earth, suggesting that they evolved first in prokaryotes and have since been adopted by eukaryotes. This, again, is evidence for evolution. Cell signaling in both prokaryotes and eukaryotes occurs via signal transduction pathways. So what is that? Well, a signal transduction pathway is a series of steps in which a signal from outside the cell is converted into a specific cellular response. So signal, transduction, response. Okay, so that cell signaling occurs in 3 stages. We have the signal, but the signal has to be received by the cell. So that's reception. So signal, reception, transduction, and then finally the response. Now the transduction, as you can see here, often takes many steps, and often that response is amplified. So one signal molecule can create a very large response in a cell, okay? So let's look at the first step first, signal reception. A signal molecule that produces a response in a cell is called a ligand. There are all sorts of ligands, okay? Cells affected by a specific ligand are called the target cells. And a ligand binds to a very specific receptor on the target cell. So again, just like when we looked at enzymes, there has to be an exact fit, has to be a match. Okay, so the ligand and the receptor have to be a match on the target cell. Only when the necessary receptor proteins on the target cells are present can the cell respond to the signal or ligand. Okay, so you can see here, here's the cell that sends out the signal. Here's the signal or ligand. Okay, this cell has a receptor that matches this ligand, so that cell will have a response. This cell does not have a receptor for that ligand, so no response. Okay, signaling begins with the recognition of that chemical message, the ligand, by a receptor on the target cell. So the ligand binding domain, meaning the part of the receptor that actually is able to bind with the ligand, recognizes that specific chemical messenger. Now that chemical messenger could be many different types of molecules. It may be another protein. It may be a small chemical. It could be a peptide. It could be a variety of things. Okay, but it's something that's going to match that protein exactly. So here we have the ligand coming over. Here's the area on that protein where the ligand can bind. Here's the receptor. And then here is the ligand bonded to the receptor. So that's the receptor ligand complex, just like we typically have the enzyme substrate complex. Now, the signal reception is non-covalent and reversible. Why would it need to be reversible? Well, cells need to be able to stop responding to a signal after the appropriate response has occurred. So if it wasn't able to reverse it, the cell would not be able to stop responding. Okay. Signal transduction pathways often involve allosteric regulation, which is when a protein's shape changes in one area, often the active site, as a result of a molecule binding elsewhere on the protein. So the ligand is binding, right, to that in the model that we showed before, to a protein and an area outside the cell, but maybe an area of that protein inside the cell changes shape. Allosteric, okay? Now, receptors can be classified by their location, which is what I was just talking about. So we can have intracellular receptors, and they are located inside of a cell. Their ligands typically are small or nonpolar. They're going to be able to diffuse across the membrane. So if they are steroids, for example, steroids are lipids, they are hydrophobic, and they're able to diffuse across the membrane, but other things can as well. Now, membrane receptors are located on the cell surface, and they typically have ligands that are large or polar or hydrophilic, and meaning they cannot diffuse through the membrane without help or cannot diffuse through the membrane at all. So two common types of membrane receptors are G protein linked receptors and ligand gated ion channels. Now, intracellular receptors. So again, remember, some receptors are intracellular. That means the receptor itself is inside the cell. So in order for the receptor to bind with the ligand has to cross the membrane. So small or hydrophobic ligands can readily cross the membrane and activate these receptors. Examples are steroid hormones. So that includes testosterone and estrogen. If an intracellular receptor is activated, okay, one of the things that it can do is it can act as a transcription factor, which means it can turn on parts of DNA. So DNA that was not currently being expressed can now be expressed and produce new proteins or more of a certain protein. Now we have ligand-gated ion channels. This is the other type. So these are closed channel proteins, but they're going to change shape when that ligand binds. Remember, that's talking about that allosteric interaction. And when it changes shape, when the ligand binds, it allows ions to diffuse either into or out of the cell. Remember, ions have a charge, so they're not able to cross the membrane on their own. They need that channel to be able to cross. So that change in the ion concentration inside and outside the cell can initiate that signal transduction pathway leading to a certain cellular response. For an example, acetylcholine receptors on skeletal muscle cells are ligand-gated ion channels, and they bind to acetylcholine, acetylcholine being the ligand. And when acetylcholine binds, it opens the channel and it allows sodium and calcium ions to diffuse into the cell. And this activates the motor proteins in muscle cells, that's actin and myosin, causing the muscle cells to contract. Now, we also have, remember, membrane receptors. And one of those examples was G protein linked receptors. So a G protein linked receptor is a receptor that's on the plasma membrane that works with the help of a mobile, meaning moving around, protein called a G protein. G proteins, when they're inactive, are bonded to a low energy molecule called GDP. Now, this is very similar to ADP. Okay? When GDP is replaced by an energy-rich molecule called GTP, think again, very similar to ATP, the G protein becomes activated. So what does that mean? When the G protein has GDP attached to it, inactive. But when that G protein has GTP attached to it, boom, active. The binding of a ligand to the receptor allows it to bind to an inactive G protein on the cytoplasmic side. So we have kind of this cascade that happens. It starts with this. A ligand binds to a receptor. That binding allows that protein to bind to an inactive G protein. And that causes GTP to replace GDP on the G protein. And now the G protein is active, and the active G protein goes and finds a different protein on the membrane that then ultimately begins the cellular response. It triggers that cellular response. So you have a ligand binding to a receptor. The receptor changes shape. That changing of the shape allows it to bind to an inactive G protein. That binding replaces the GDP with GTP. Now the G protein is active and it goes to a different protein, activates it, which then eventually causes a cellular response. So we can look at that here. So here we have that G protein coupled receptor. Okay, so here's that receptor. We can see the ligand binding, right? We can see this G protein is inactive, and we can see a different protein. In this case, it's an enzyme here. Once the ligand binds to the receptor, changes shape, you can see GTP replaces the GDP, and then this G protein travels over here, activates this enzyme, and now we get a cellular response. And remember, part of that is that they're reversible. So again, that GTP is going to be replaced with a GDP. This enzyme will be inactivated and it can continue. Okay, now similarities in structure among G proteins and G protein linked receptors in modern eukaryotic organisms like yeast and fungi and protists and animals and plants suggest that these G proteins and the G protein linked receptors evolved very early in life on Earth. So how do we know? Well, we look at some of the evidence. And all G protein linked receptors have 7 transmembrane domains. So look here. Go up and down and up and down and up and down through the membrane 7 times, specifically 7. Okay, so there are 7 regions that cross the cell membrane, that bilayer. And all G proteins are composed of 3 polypeptides. They have the alpha, beta, and gamma subunit. Most of the time, we're just focusing on the alpha subunit because that's where the GDP is bound. But every single G protein has an alpha, a beta, and a gamma subunit. So this, again, evidence of evolution. All right, so now I want you to take some time with your table partner and try and work through the questions related to topic 1 in your notes. What do I mean? Well, I mean here. So the ones related to topic 1, here and here. Okay, so if you're doing this on your own, pause here so you can try and get these questions answered on your own first. Now we're going to go over the answers. So first, draw a picture of cell signaling that includes reception, transduction, and response. So here's an example of a picture. So we have reception right here. We have transduction right here. That message gets relayed. And then we have response here. And here it says label the ligand and the receptor in your picture. Here's the ligand. That's the chemical that is going to bind to the receptor on the cell membrane. A cell releases ligands, but only some of the nearby cells appear to respond, while others do not appear to be affected. Why is this? Well, receptors. That's the answer. Simply, but let's give more. Only cells with the correct receptor for the ligand will respond. So if it has the right receptor, it will respond. So here's the cell that sent the signal. This cell is a target cell because it has the right receptor. This cell does not have the right receptor, so it's not a target cell. A drug was developed that inhibited all of the membrane receptors on the surface of a cell. It was observed, however, that estrogen hormones still had an effect on the cell. How is this possible? Well, think for a minute. If estrogen still had an effect, that must mean that estrogen's effect is not related to membrane receptors. Because if all the membrane receptors are inhibited, they wouldn't be working correctly, right? Well, estrogen is a steroid hormone. That means estrogen is a lipid and nonpolar and hydrophobic. So it can cross the cell membrane. Estrogen has an intracellular receptor, so it's not embedded on the cell membrane. So because only the cell membrane receptors were affected, it would not affect estrogen having a response because it has an intracellular receptor. In general, what type of ligands do not bind to membrane proteins? Do these messengers have receptors? If so, where? Well, that would be nonpolar ligands, things that are hydrophobic. Why? Because they can cross the membrane, and they would have intracellular receptors in the cytosol. So that's the image that we just looked at. Instead of binding to a receptor on the cell membrane, they're going to bind to an intracellular receptor. The ligand-gated ion channel pictured below is found in the membrane of a neuron or nerve cell. The membrane of nerve cells also contains a lot of sodium-potassium pumps that produce high Na concentration plus concentration outside the cell and a high K plus concentration of ions inside the cell. Given this information, what do you think is the identity of the small dots? So let's look at it. We have a lot of these dots outside, and then they're moving inside. So I can tell from this picture that there's a lot of them on this side. Well, this says it's the cytosol on this side. So that means this is outside of the cell, right? So which concentration is high outside the cell? Yeah, Na plus. So why? The NaK, sodium potassium pump, creates a high concentration of Na plus outside the cell. So here's the sodium potassium pump, and it pumps the NAs out of the cell and Ks in the cell. Okay, that's maintaining the high Na outside, so then it can diffuse through a ligand-gated channel right here. So here just is another look at this is a sodium potassium pump, and it's going to pump out 3 sodiums for pumping in every 2 potassium. This is going to require ATP in order to keep that sodium concentration high outside the cell. Okay? Whereas potassium is going to be high then inside the cell. And when ligands bind to those channels, those ions can diffuse across. Here's just another model of it. You can see that it's 3 sodium. It requires ATP, 3 sodiums out, and then 2 potassiums in. Draw a picture of a G protein linked receptor, a G protein, and another membrane protein that is activated by the G protein. So here's a model. So here we have a receptor. Here we have the ligand, which is a hormone, but just a ligand that works with this receptor. Here's the G protein, and here's another membrane protein, which in this case has an E representing an enzyme. When the hormone binds, this receptor changes shape, causes the G protein to bind to the receptor, which causes the GDP to get replaced with a GTP. This becomes active and moves over in the membrane and binds with this enzyme and activates it. Remember that every receptor that is links to a G protein has 7 sections that cross the membrane, and every G protein, right, that binds with this receptor has an alpha, a beta, and a gamma subsection. The alpha subsection is the one we focus on because that's the one that has GDP bound to it. Whew, that's the first section.